Something that we all struggle with is clinical trial enrollment. We haven't reached our goals in terms of getting a number of patients that we would feel comfortable with in terms of trial enrollment. So it's estimated that about 5 to 7% of U.S. adults with cancer will enroll in a clinical trial. And that has not changed significantly over time, despite a lot of our efforts to increase trial enrollment. And there's a lot of different factors that play into that. Some of them are institutional, some of them are patient understanding of trials. They come from a lot of different directions. But one thing that we know is that trial complexity can play into that decision for patients. And over time, we've seen clinical trial complexity increase. And that has come with the paradigm shift within oncology from a focus on chemotherapies to targeted therapies, immunotherapies. And so the purpose of this study was to really understand how we could quantify time in the clinical trial setting. Time toxicity is not a new concept. This is something that has been quantified there have been attempts to quantify this in the clinical setting as a way of communicating with patients, informing patients about the time requirements of different treatment regimens. However, it's something that's studied in the clinical trial setting. And so the purpose of our study was to see if we could identify a mechanism to quantify time in the clinical trial setting.